Welcome learners. Today's topic to learn is G protein coupled receptor and CAMP pathway. G protein coupled receptors GPCRs were named for their common ability to associate with heterotrimeric G proteins. GPCR or cell surface receptors structurally composed of three domains, namely an extracellular amino terminus, seven transmembrane alpha helical domains, and intracellular carboxy terminus. GPCRs recognize a variety of signals ranging from photons to ions, proteins, neurotransmitters, and hormones. The GPCR superfamily comprises at least five structurally distinct subfamilies: glutamate, rhodopsin, adenosine, frizzled or taste2, and secretin receptor families. About 90% of all GPCR belong to the rhodopsin family. The human genome encodes nearly 800 GPCRs, representing over 3% of human genes. G protein. G protein is a guanine nucleotide binding protein that can interact with a G protein linked receptor. G proteins are associated with the cytosolic side of the plasma membrane, where they are ideally situated to interact with the cytosolic tail of the GPCR when a signal binds to the GPCR. G proteins are heterotrimeric in nature and they have three subunits: alpha, beta, and gamma. The alpha subunit binds GDP in inactive state and exchanges GDP to GTP in active state on ligand binding. Knowing about the GPCR signaling. The binding of a signal molecule by the extracellular part of the G protein linked receptor causes the cytosolic tail of the receptor to interact with and alter the conformation of a G protein. This has two consequences. First, the alpha subunit of the G protein loses its GDP and binds to a GTP instead. Second, the G protein breaks up into GTP bound alpha part and the beta gamma part. The next step in GPCR signaling is activation of adenylate cyclase. The free GTP bound alpha subunit then binds and activates adenylate cyclase. Activation of adenylate cyclase involves CAMP production. Adenylate cyclase is an enzyme which converts ATP into cyclic AMP, which is a second messenger. The structure of ATP and cyclic AMP you can see in the slide. Signal transduction. After the CAMP production, which is the second messenger, CAMP binds to and activates other downstream targets such as protein kinase A, which further phosphorylates other proteins in the signal transduction pathway. Here in the picture you can see the protein kinase A, which has two catalytic subunits and two regulatory subunits. After the release of CAMP, the CAMP binds to the catalytic subunit and releases the regulatory subunit. Binding of CAMP to the catalytic subunit is considered to be the activation of protein kinase A, which succeeds in phosphorylating the other signal transducing proteins. So the phosphorylated proteins include enzymes involved in glycogen breakdown, enzymes involved in muscle contraction, transcription factors, ion channels, other kinases, and receptors. Receptor deactivation. After enough cellular effect, the receptor has to be deactivated in order to maintain the homeostasis. Receptor deactivation is a crucial process that prevents excessive signaling and allows for receptor recycling. The G-alpha subunit has intrinsic GTPase activity, which means it can hydrolyze GTP to GDP. This hydrolysis inactivates the G-alpha subunit, causing it to reassociate with the beta-gamma subunit dimer, reforming the inactive heterotrimeric G protein. This slide shows about the diversity of GPCR signaling mechanism. We know that G protein consists of G alpha, beta and gamma subunits as they are heterotrimeric proteins. The G alpha subunits have been classified to have four subfamilies. They are G alpha S, G alpha I, G alpha Q, G alpha 12 based on structural and functional similarity. We know that G alpha S leads to the production of CAMP, whereas G alpha I belong to a family which inactivates the activity of adenylate cyclase and decreases the CAMP production, whereas G alpha Q is a family which activates phospholipase C and ultimately leading to the intracellular calcium mobilization from the endoplasmic reticulum. G alpha 12 family is involved in inactivation of Rho family of small GTPases. Thank you all.